Hello, this is Delanie Dantuma, Nick Fabbrocile, Olivia Nguyen, Lauren Winkowski, and Maria Alvear. Today we are going to show you the RTK-PI3K AKT pathway. The origins of PKB AKT research can be traced back to the discovery in 1977 by Stahl and co-workers with phosphatidylinosole 3 kinase isolated in 1990 by the group of Cantley. In 1995, Richard Roth and his colleagues reported that AKT was activated by insulin. Current research has been focused on how the pathway relates to developmental and overgrowth syndromes, cancer, cardiovascular disease, insulin resistance, type 2 diabetes, inflammatory and autoimmune disorders, as well as neurological disorders. This pathway is important in signaling cell cycle progression, cell survival, glucose metabolism, apoptosis, and protein synthesis cell growth.
RTKs are cell surface receptors that recognize many polypeptide growth factors, cytokines, and hormones. This receptor has 3 functional domains: an extracellular ligand binding domain, a transmembrane domain, and an intracellular tyrosine kinase domain. When a ligand, such as a growth factor, binds to the RTKs, the 2 RTK monomers form a dimer. This conformation change leads to the activation of intracellular tyrosine kinase domain and autophosphorylation by each monomer.
After the conformation change of RTK-PI3K is recruited. PI3K consists of 2 domains, a catalytic domain P110 and a regulatory domain P85. The activation of PI3K occurs as a result of the regulatory subunit binding to the activated receptor or indirectly activated via adapter molecules such as the insulin receptor proteins. PI3K can also be activated by GTP binding RAS protein. PI3K is a kinase capable of phosphorylating the 3-position hydroxyl group of the inositol ring of PIP. Phosphorylation will transform PIP2 into PIP3. PIP2 and PIP3 are minor phospholipid components of cell membranes. The PIP3 usually serves as a docking phospholipid that binds specific domains that promote the recruitment of proteins to the plasma membrane and subsequent activation of signaling cascades.
Regulatory molecule phosphate and tinase humanolog will downregulate the pathway by converting PIP3 back into PIP2. After PIP3 is created, it will recruit AKT. AKT is a serine threonine specific protein kinase that plays a key role in multiple cellular processes. Once bound, AKT will transform into protein kinase B. Once activated, it will regulate the function of a broad array of proteins via photophosphorylation. Examples of AKT inhibitory proteins include protein phosphate 2A and phosphate PHLPP.
Protein kinase B, or AKT, will be activated via phosphorylation by PDK1. As previously stated, ATK has the capability to translocate to different parts of the cell and regulate many different proteins. In this mechanism, AKT will dissociate from PIP3 and PDK1. AKT will then bind to active BAD, leading to BAD inactivation. BAD is a protein that, when oligomerized, leads to the release of several apoptogenic molecules from the mitochondrion. The inactivation of BAD will in turn activate BCL2 and lead to the final result of preventing apoptosis.